In the previous video, we looked at how an action potential can be conducted from the trigger zone near the beginning of the axon down to the synaptic knobs. In this video, we're going to look at how the action potential is actually transmitted from one cell to the next cell. That could be from one neuron to another neuron, or it could be from a neuron to an effector organ, such as a skeletal muscle fiber. But before we do that, let's go ahead and review first the all or none law, and then we'll take a look at the refractory periods again. In the all or none law, we see that if a stimulus is subthreshold, we won't get any action potential whatsoever, all or none. But we also see that action potentials are all approximately the same size, regardless of how strong a superthreshold stimulus is. Here we can see a stimulus that gets us past threshold, and we end up getting 4 action potentials that are all approximately the same magnitude. When the stimulus is even greater, the magnitude of the action potentials is no greater. They still go to approximately plus 30 millivolts. It's just that now, within a given period of time, we get more action potentials. And so again, we see the all or none law. Another important thing about action potentials is that we end up having what are known as refractory periods. There are 2 of those. The absolute refractory period means that no matter what we do during this first millisecond, we can't get a second action potential superimposed upon the first one. The reason for that is that, at least initially, we've opened up pretty much all of the voltage-gated sodium channels. We can't open up any more than all of them, and so we're not going to start a second action potential. For the second half of this, during the repolarization phase, the inactivation gate of the voltage-gated sodium channel is closed, and there's nothing we can do other than just wait until it opens up again. As we progress through the action potential, we get into what's referred to as a relative refractory period. During the relative refractory period, we could superimpose another action potential here, but it would require a stronger stimulus than what is typically used. And here you can see that the membrane is actually hyperpolarized, and you see that it's further away from threshold than it is at rest, and so it takes a stronger stimulus to get us to threshold and get a full-blown action potential. Those are what are known as refractory periods. Again, the action potential is able to travel from the trigger zone all the way down to the synaptic knobs. It's important that you understand that an action potential doesn't travel anywhere, but one action potential creates a second one in the adjacent piece of the axon, and we can see why that is, because here, the way the charges are lined up, we get this flow of current, and the flow of current through the piece of membrane that is at rest is going to open up voltage-gated sodium channels, creating an action potential here. And that'll create a third action potential a little bit further down the axon, which will create a fourth further down the axon, and so on until we get all the way down here to the synaptic knobs at the very end of the telodendria. And then again, as we've said, not all synapses between neurons are from axon to dendrite, although many can be. We can also have those that are from axon to axon, so axosomatic. We can have those that are from dendrite to dendrite, and we can even have those that are from axon to soma. And again, here you can see a different drawing that perhaps shows that a little bit more clearly, because we see more of the neurons involved here. And so here's an axosomatic synapse, here's an axonic synapse, and here's an axodendritic synapse, which is what we most commonly think of a synapse being. And here you can see a micrograph of some axosomatic synapses. There's the axon of one cell synapsing with the soma of a second cell, and then again axosomatic. Now we need to talk about how an action potential is transmitted from one cell to another cell, and the fastest way for that to occur is for it to occur electrotonically as opposed to chemically. An electrotonically transmitted signal uses protein channels known as gap junctions, and so here you can see those gap junctions in the membrane. The gap junctions essentially connect the cytoplasm of one cell with the cytoplasm of another cell. And so here we're in the cytoplasm of cell 1, here's the plasma membrane of cell 1, and here you can see that protein channel. It lines up with the protein channel that's in the plasma membrane of cell 2, and so now we can have flow of ions directly from cell number 1 to cell number 2. And here you can see the diagram from your textbook illustrating gap junctions. Gap junctions are found throughout the body. We do find them in the brain and neurons, but they also connect astrocytes to one another in the brain. We find them in cardiac muscle, smooth muscle, the liver, and the pancreas. And here you can see gap junctions that connect smooth muscle so that all of these smooth muscle fibers contract very rapidly, one right after the other. The diagrams that you see here don't do a very good job of indicating the concentration or the number of gap junctions that we have in a particular area. And here now you can see that much more clearly. There are just way more gap junctions than you see here in the diagram. And so this clearly is going to allow for transmission of ions much faster from one cell to the next cell. Much more commonly, we find chemical synapses, and so the signal has to be transmitted chemically from one cell to another cell. And here we can see a presynaptic neuron. And this picture from your textbook is simply showing release of neurotransmitter from the presynaptic cell. And you can see that what happens is as the action potential arrives, it's actually going to excite these other voltage-gated channels, which are now voltage-gated calcium channels. And you can see that those are found throughout the membrane in that synaptic knob. And as these voltage-gated calcium channels open up, calcium will enter the synaptic knob. And calcium is a very powerful and very potent intracellular signal, and it's going to signal both migration and docking of these synaptic vesicles to the membrane down here at the bottom of the synaptic knob. One thing that this doesn't show you is that the synaptic knob is actually crammed full of synaptic vesicles, these little phospholipid packets that are full of neurotransmitter. And then as the vesicles fuse with the membrane, they'll open up and they'll release neurotransmitter via exocytosis, which again is an active process and which does require the use of ATP for this to occur. Here you can see a slightly better depiction of the end of a synaptic neuron. And because exocytosis is active, we need lots and lots of mitochondria, and we also are going to have a lot of synaptic vesicles in here. And so again, this explains why we get this swelling at the very end of the axon and why we have this synaptic knob. What this picture doesn't show you is what's happening on the postsynaptic side. And so we do have the membrane over there, and you should be aware that transmitters can't do anything without receptors to receive them and to be activated by them. And so here in this picture we can see these integral proteins, which are both receptor for the neurotransmitter but also channel for ions, and these now represent ligand-gated ion channels. Ligand is a chemical or a chemical signal. And so the chemical is the neurotransmitter. It has to bind with the receptor, which is a part of the ligand-gated ion channel. And then that channel will open up, and you can see that since it's a ligand-gated ion channel, we get some sodium coming in, but we also get some potassium leaving. The important thing is that we get more sodium coming into the cell, and so that's going to cause depolarization of the postsynaptic membrane. Here again, you can see the presynaptic side of the synapse, and there you can see a lot of synaptic vesicles in there, and you can even see some neurotransmitter being released, and it will diffuse across the synaptic cleft to get to the receptor. Here you can see a different depiction of a chemical synapse. This one happens to be the chemical synapse that links an alpha motor neuron with skeletal muscle membrane, or the sarcolemma. And they've added one more piece for us. Here you can see the voltage-gated calcium channels, which are opened up when the action potential reaches the synaptic knob and calcium enters. You can see that this thing is crammed full of synaptic vesicles. The calcium causes migration, docking, and fusion of the synaptic vesicle, and then it opens up and the neurotransmitter is released via exocytosis. Now, the human phys book that we use, it presents a lot of detail as far as fusion and docking is concerned. And I don't want you to get into that. You need to check with your professor to see how much detail she wants you to understand in terms of the complex process of docking and fusion of those synaptic vesicles. And here you can see that the neurotransmitter will in fact diffuse across the synapse, and it will bind with the receptor sites on these ligand-gated ion channels. Now there's something that I want you to understand. The neurotransmitter does not cross the postsynaptic membrane and get into the inside of the cell. What does that is the ion, and typically it's the sodium ion. And so sodium moves across and causes a depolarization here. And it's that depolarization which opens up these voltage-gated sodium channels that are located either at the trigger zone of another neuron or here outside of the neuromuscular junction for a muscle cell. And so when we open up a ligand-gated ion channel, what we do is we cause a little bit of a depolarization. And this depolarization is referred to as an EPSP. And again, many of you now know that EPSP stands for an excitatory postsynaptic potential. It's different than an action potential. It doesn't always get us all the way to threshold. Here you can see this one depolarizes us to approximately -60, -58, somewhere around there. And then the sodium-potassium pumps quickly return us back to the resting membrane potential. These EPSPs are typically evoked by a neurotransmitter such as glutamate in the central nervous system. Glutamate is the most important excitatory neurotransmitter in the central nervous system. Or it could be acetylcholine. Acetylcholine is the most important excitatory neurotransmitter in the peripheral nervous system. And so it's this neurotransmitter binding with the receptor which alters membrane permeability by opening up the ligand-gated ion channels. And here we can see a little bit of a detail of a ligand-gated ion channel as the neurotransmitter binds here. The channel opens up, sodium diffuses into the cell, potassium diffuses out, and while you might think that those two things would balance each other out, there is a stronger force acting on the sodium, and so more sodium enters the cell, and we do get a depolarization or an EPSP. We can also actually make the membrane more polarized, in which case we get an IPSP, and you might guess that I in this case stands for inhibitory, and that's exactly what it means. It's an inhibitory postsynaptic potential. IPSPs are evoked in the central nervous system by either GABA or glycine. And then in the peripheral nervous system, unfortunately, they can also be evoked by acetylcholine using a different type of acetylcholine receptor. And here we can see the way that a GABA receptor will work. And so in this case, it actually takes two molecules of GABA to bind with the receptor. And then again, the ligand-gated ion channel opens up. But now, rather than having sodium enter the cell, we have chloride enter the cell. Chloride is bringing in a negative charge, and that's what's responsible for the hyperpolarization that will occur there. Here you can see the EPSPs. This is the figure from your book. Another important thing about EPSPs is that two EPSPs happening close together, either close with on the surface of the neuron or close together in time, can actually add together. This second EPSP is adding on top of the first one, and while each EPSP alone is not enough to get us to threshold, two of them together do get us to threshold, and so we do indeed get our action potential. And here we can see an example of a nicotinic cholinergic receptor. The neurotransmitter that activates this channel is acetylcholine. It takes two acetylcholine to open up a nicotinic cholinergic receptor, but once we have two of them, the channel opens up. Sodium then enters the cell. Potassium leaves the cell, but again, we have more sodium entering the cell, then leaving. And here we see our IPSP. In the peripheral nervous system, an IPSP can be created by acetylcholine, but now you can see that it works a little bit differently. The acetylcholine in this case is going to bind to the receptor and activate something called G protein. Your textbook talks about G protein. The G protein is going to dissociate, and part of it moves across the membrane. Remember, phospholipid bilayer membrane is very fluid, and so this protein moves through and it opens up a potassium channel. Potassium now goes down its concentration gradient, taking positive charge with it, which can also hyperpolarize the membrane. Again, look, we're further away from threshold now than we were before, and so it's going to take a larger EPSP in order to reach threshold and get an action potential. This diagram also shows how both EPSPs and EPSPs and IPSPs can add together and either create or not create an action potential. And that brings us to the end of this particular slide, this video.